What I present was the very sort of initial experience on how to set up the platform, but I concentrate more on the clinical development area, which is centered around a platform style, what we call master protocol, but really it's a portal where we can approach the patient to who we think are suitable for our clinical trials so that we can biopsy the patient and perform molecular profiling for the patient and then deliver a patient to one of the downstream clinical trials. So I presented our initial experience on both along the patient pathway as well as the patient tissue, the tumor tissue processing pathway, and demonstrated what are the sort of challenges and how we overcame it. But again, being the disease itself, pancreas cancer itself is, unfortunately, patients' performance status can deteriorate fairly rapidly, so that it can, patient does, unfortunately, even though when we first approach a patient, it's only a few weeks later that when we are ready to start the treatment, patients' performance status declines so fast that they're no longer ready for treatment. So there are a lot of things that we've learned and we can, do to improve on shortening the time from diagnosis to treatment, as well as not just the patient pathway, but also the tissue pathway as well. And we did a lot of sort of troubleshooting, protocol optimization on how to best acquire the tissue and how to best do the molecular profile of the tissue. Because in general, pancreas cancer is a very highly stromal type of cancer, meaning that there's a lot of fibrotic tissue, and then they will have islands of cancer tissue, which means it makes it difficult for generation sequencing technology to detect mutations. So we did do a lot of sort of background homework as well on how to accommodate for formalin tissue, which is clinical grade tissue, as well as low input, as well as low cellularity in the input material. So that's why I shared, but it is achievable. We just had to do quite a bit of homework and a bit of coordination.